Neurogenesis is the process through which new neurons are generated in the brain. In humans, similar to animals, this process occurs in a particular region called the hippocampus. Evidence from research conducted in animals shows that stress can decrease hippocampal neurogenesis, and this is associated with reduced memory and learning and low mood. This is similar in humans. For example, when you are stressed, you may have noticed experiencing a lack of attention, problems with remembering things, and in some cases, even depression. Altogether, these cognitive and mood symptoms may be associated with a reduction in hippocampal neurogenesis, although this link is still indirect and difficult to test in humans. Considering this, we have decided to focus our investigations on animals and summarized evidence showing changes in hippocampal neurogenesis, cognitive function, and behavior in animals exposed to stress during adolescence. In particular, we found that animals exposed to stress show a reduction in hippocampal neurogenesis, memory and learning, and an increase in depressive behaviors. Moreover, some studies have also shown that these alterations can last until adulthood. But how can we prevent this from happening? Interestingly, some studies in adolescent animals show that antidepressants or a healthy diet, consisting of omega-3 fatty acids and vitamin A, can reduce the negative effect of stress on neurogenesis, cognition, and behavior. Accordingly, evidence in animals has demonstrated that antidepressants require an active hippocampal neurogenesis for them to have beneficial effects on both cognition and behavior. However, evidence generated so far is relatively limited and mainly focusing on adult animals. Therefore, future research should aim to investigate these changes in neurogenesis also in adolescent animals and then to replicate these findings in adolescent humans.